I hear that we are live, so that means I can formally welcome you to this very interesting and special evening, the Kafatos lecture, which will be given by Hans Klevers. And I have only a few minutes, but there's lots of connections, of course, between Hans Klevers and this building. My name is Marleen Dochtrom. I'm a professor at the Technical University of Delft, but here now in the role as president of the Royal Netherlands Academy of Arts and Sciences. And this is our house. So welcome to our house. Our academy exists since 1808. So we were started by King Napoleon, Lodewijk Napoleon, who gave us the academy. And since 2005, we're also the home of the Young Academy. And what's interesting is that our academy holds all domains of science, so medical sciences, technical sciences, but also social sciences and humanities. And over the years, we also have presidents from all these different backgrounds. Actually, 10 years ago, Hans Klevers was our president, and a few interesting things happened, I think, during that year. We started, for example, also the Academy of Artists. So when Napoleon started the academy, we were sciences, humanities, but also the arts. And then quite quickly in that same century, the arts were kicked out. And in 2014, we got them back under your leadership. The Academy has several roles. Not only are we an advisor to our government, we are, of course, very interested in the public debate about science. So today's lecture is really a prime example of that. And we are responsible for 10 research institutes, one of them being the Hubrecht Institute. And besides being our president, Hans was for many years the director of the Hubrecht Institute. And I want to maybe say one thing about our role, not only to enhance the interaction between the public and science, but also between science and our parliament. And I have one picture that I took also from 10 years ago. I don't know if you remember, Hans. Do you remember what it is? So this is the instrument called Parliament and Science. So this is something that we started 10 years ago formally, which is where we offer members of our parliament the opportunity to interact with experts from science, all fields of science, to help them do their parliamentary work. And it was a collaboration between the Young Academy, the Academy of Sciences, and the universities. And 1st of October 2014, so exactly 10 years ago, this picture was taken in our parliament with Hans there on the side. Anyway, we are very proud to host the Kafatos lecture here at the Academy building today. It's an initiative from the alumni of the EMBL, so it's also a bit mine because I just explained. I spent, when I was 23 years old, 3 months at the EMBL. I'm a physicist by training. But at the EMBL, I learned for the first time about biology and about doing experiments, and it changed my life. So another reason that I'm very happy to welcome you all here, and all means the room here is full. But 2 days ago, I heard that there were 764 participants in total registered, but by now I think it's over 900 already. So there's a lot of people from all over the world, in fact, that are joining us today online. So welcome, I think it's over there, to all of you, and enjoy the evening. And then I'm going to give the floor to Ian Matai, one of the former directors of the EMBL, and also the chair of the selection committee. And the floor is yours.
So, good evening, and it's very nice that those of you who are here in person could come, and it's very nice that those of you who are here virtually could also join us. And I see some familiar faces, some that I haven't seen for a number of years, but it's very nice to see you again. I've been given the impossible task of introducing Hans, introducing Fotis Kafatos, introducing the Kafatos lectureship, giving thanks to all the people who need to be thanked in 5 minutes. So I can tell you, I wouldn't even be able to list the awards which Hans has received in 5 minutes. So this will be a very brief overview. It will go over time, I'm sure. but I'll try that it doesn't go over time too much. And I'll start by talking about the award lectureship, the Kafatos lecture. It was introduced by the Alumni Association, who decided to do this in honor of Fotis Kafatos, who was my predecessor as Director General of the MBL in 2021. And the idea was to choose a very good scientist who was doing science that was of societal relevance and to try to help bring this work into the public domain and into the public attention. The speakers are asked to make their talk accessible to everybody. We'll see how if Hans does this, but that's what they're asked to do. And therefore, I'd like to encourage all of you, also those of you online, to share the link. The lecture will remain online for as much as we know forever at this point. And it may well be a lecture which will be a lot of fun, for example, for advanced school classes, etc. So please spread the word. Who was Fotis Kafatos? So Fotis Kafatos was born in Crete, in Greece. He grew up on the island and went to school there. He had the luck at school to meet a very smart school teacher who realized that Fotis was capable of greater things. And so he helped Fotis to find a benefactor who funded Fotis to go at a very young age to Cornell University. So he left Crete to go to university. He was very successful as a student and as a PhD student also, then went to Harvard. At Harvard, he became the youngest ever professor at Harvard at the age of 29 years. He went on to be the head of the Department of Genetics, and again was greatly successful. He studied some of the genetics and the early molecular genetics of insect development and did a number of things demonstrating conservation of developmental mechanisms. He, at the same time, founded Forth, a research institute which still exists on Crete and for many years has been a very successful research institute. He did lots of other things in Europe and in the US, which resulted in him being recruited to be the Director General of EMBL, the third Director General of EMBL. He was Director General from 1993 to 2005. And towards the end of this time, from roughly 1998, 1999, there was a move within the scientific community to set up the European Research Council with funding from the European Commission. And this was a difficult task. There was a lot of political and lobbying effort. Fotis was supremely talented in lobbying and had the good fortune that the head of DG Research was also Greek at that time, Achillea Mitso. And he and Fotis originally didn't get along and disagreed violently on the subject of the ERC, but Fotis was nothing if not persistent, and he spoke to him. I remember at one period he telephoned him every day for 2 months to try to persuade him that the EC should be funding the ERC, and eventually it worked. And in fact, Mitso became the internal supporter of the ERC. So Fotis then went on to chair the ERC Scientific Committee and be the first president of the ERC from 2005 to 2010. So that was Fotis. Now I will thank all the people who have been, or at least some of the people who have been involved. We thank very much the Academy for allowing us to host Hans's lecture here. He chose, the speaker chooses where they would like to give the lecture. Hans chose the Academy, and they've been extremely helpful, particularly, I'm told, Hans Sittrop, who has done all of the logistic organization here. Marilyn, thank you also for the introduction to the Academy and to the event. After Hans speaks, if I ever end my introduction, Agnes Smolensky will run the question and answer session and will also provide some information on next year's Kafatos lecture. I'd like to thank the EMBO Photo Lab as well as the personnel of the Academy for all of the audio visuals, all of the technical setup, etc., which is never difficult to do when you're moving something around. What else? Finally, and most profoundly, I'd like to thank Mirnush Reiner, who is the head of alumni relations at EMBL and is tirelessly enthusiastic. It's actually tiring to work with Nush because she's so tirelessly enthusiastic about things. And the people who work with her, some of whom are here, for all of their work. And I'd finally thank the fellow members of the selection committee. Before moving to Hans, I will tell you about the Bodo Saki Foundation, which helps to fund the Kafatos Award Lecture. They are a foundation based in Greece. They help to promote educational initiatives, healthcare initiatives, environmental initiatives, and they also promote the distribution of science in civil society. And we thank them very much for the support that they've given. So now I come to Hans, and Hans, unfortunately, I only have 30 seconds left, so you will have to introduce yourself. I'm sure many of the people in this room know Hans as well or better than I do. He studied in Utrecht. He worked in Utrecht for, I don't know, 57 years or something. You can list all of his positions in Utrecht. In between, he went to the Dana-Farber Cancer Institute in Boston to work with Coax Terhorst, as I don't know if it was a PhD or a postdoc, but he worked for a few years in Harvard. He came back to Utrecht, started to work on signaling of a growth factor, Wnt, which particularly initially, I think in flies, where it's called wingless, was shown to have very important roles in early development. And Wnt also has roles in development in many other organisms. Hans was involved in showing that Wnt signaling was also very important in certain types of cancer, and then went on later to show that in the stem cells of the epithelium of the gut was also important for stem cell growth and renewal. He got very interested in these stem cells and actually worked on the gut epithelium for many years. And eventually, having identified the stem cells, wanted to grow them. And so he collaborated with Sato in Japan, who had worked out how to grow cells in collagen matrices, to grow the cells in 3D. And I think, to their surprise, when they did this, they didn't just grow and divide, but they actually differentiated and started to make something that looked like gut epithelium. At the same time, Sasai, again in Japan, was doing similar experiments with induced pluripotent stem cells and making similar structures of different types of tissue from those cells. And so those two, Hans and Sasai, really made the first of what are now called organoids. I don't know where the name came from, but maybe Hans will tell us. Organoids are obviously a complicated system, but they're not as complicated as human beings. And so they're a good potential model system for studying aspects of human genetics, of testing drug effects, testing the effects of mixing different types of cells, et cetera, et cetera, et cetera. The potentialities are large, not infinite. They're not as complex as human beings, but they're on the way to being that. And as a result of this, and actually his earlier work on cancer, Hans has a huge number of awards, none of which I'm going to talk about. Recently, 3 years ago, Hans moved himself, but not his lab. His lab is still in Utrecht in 2 pieces, to Basel, where he became, he has a very corporate set of titles that would take longer than 5 minutes to list. But he's basically head of global research at Roche. And tonight, I hope he's going to tell us what his lab and what research at Roche is currently doing on the basis of his discoveries. I'm very jealous of the fact that he obviously has access to infinite AI and computational resources, because when we asked him where he'd like to give the talk, we also asked him to pick a date. And he already knew more than a year ago that there were going to be 3 sunny days in Amsterdam in October in a row. And he chose those dates to give his lecture. Hans, please. So there is a Tommy certificate which goes with the award to remind you of it.
We need
Yeah. And there's also a rendering by Combs in EMBL of a brief description of some of the research.
That's
done.
also on the poster. Right.
And it's also on the poster. So,
All right. Thank you very much. Yeah, well, Ian, thanks very much for all this introduction, and also, of course, to the EMBL, the EMBO alumni organization to invite me, I guess, to give the second Fotis Kafatos lectures, a third. It's a great honor. What I'll try to do, you already give away a little bit, but I'll try to give you some background on how we sort of stumbled across our version of organoid technology, and I'll try to do it in a way that is accessible to also the deep biomedical scientists that are here, but also people who are interested in science but might not understand all the little details. So there'll be a lot of little stories, and I hope that I don't lose you. And there's a lot of moving things on the screen as well. That also in my experience helps to keep the attention of the people. So what you see here is the insides of the gut. I'll first say a little bit more about stem cells, but then I'll return to this particular organ that we sort of discovered halfway my scientific years as being an extremely exquisite organ to understand how stem cells work. A little bit about stem cells. So aging, being in the Royal Academy, and we have a few good examples in the audience, and I would probably feel that I would be in that class by now. So what essentially happens is over time, our bodies get born, they get formed during fetal development. We're born, we grow, probably somatically our peaks are in our 20s, early 30s, and then slowly but steadily our body decays. We lose, so we consist of cells and of organs, tissues. We live much longer than most of our cells. So skin cells, maybe a year or so, hair cells, you can actually see the stem cells producing hair every day. Your nails grow out, you cut them. This all because cells are born from stem cells, they work hard, and then a little bit later when they're worn out, they die and they get replaced. So nature has invented stem cells. This is I believe, a liver stem cell. So what stem cells do is quite dumb. They just divide. And whenever new cells are needed, they just divide enough to fill up a gap that would otherwise form in the tissue in which they reside. Now, there are 2 types of stem cells. The types of stem cells I'll be talking about are what we call the somatic stem cells. So the cells that you carry around in all of your organs. almost all your organs, that actually continuously replace worn out cells. And when there's damage or when there's disease, they probably work a bit harder. The fact that you age is because the stem cells themselves also age. For mice, this happens in 3 years. For humans, 80, 90 years or so is probably the average of a normal human body. When things go wrong, when stem cells become overactive, this is, and it happens by mutations, for instance, this is what leads to cancer. Stated in a simple way. Now, what happened, this was probably in the mid-90s. We had indeed been interested in Utrecht in trying to understand how T lymphocytes form. We then uncovered a part of the Wnt pathway, originally defined by Roel Nusse, on fly work and mammalian work on the windlass Wnt pathways. Studied almost entirely by developmental biologists, so people interested in fly development or frog development. We, by chance, discovered a key component of this pathway, gene, TCF we called it. We knocked it out. There were 4 of them, TCF4 when we knocked it out. What you're looking at here is the inside of the gut. This process that you see came to an acute halt. And what are you looking at? This is actually a gut, so food would pass by from left to right. The surface of the gut has these extensions, they're called villi, or in Dutch, darmvlokken. They make sure that the uptake of nutrients becomes very efficient. You need a large surface, like in the lungs. Your surface is very large to take up oxygen. Here's nutrients. These cells are exposed to the insides of your gut, probably the most threatening biological environment on this planet. They have a very short half-life. They are born at the base of the crypts, and this was all work done in the 50s and the 60s of the last century and then sort of forgotten. When we started reading about this, it turned out that there must be stem cells somewhere near the base of the crypt. Down here, you can see my cursor. Daughter cells divide very rapidly, so they could be easily seen on a microscope. The stem cells were not really easily identified. After 2 days, so they divide 4 or 5 times, they leave the crypt of Lieberkühn for the insiders. They then rapidly develop into one of about 10 different cell types that you need to digest food, to take up the nutrients, but protect yourself against the bacteria that are in there. But also, for instance, the now very popular weight control drugs, the GLP-1 drugs. It's actually a hormone made by one of the 6 cells in the gut that make hormones that control how much we eat and how our metabolism is regulated. So once they're formed, 2 days after they're born from these enigmatic stem cells, they then together keep on moving up. Now they work very hard. They're now exposed to the lumen of the gut. They try to pump nutrients and liquids into the body, into these blood and lymph vessels, while they move up. And then when they're 5 days old in the mouse, humans probably about 7 days old, they reach the tips of the villi and they commit suicide, called apoptosis for cells. So as you sit here, probably every week you renew your entire lining of the gut. The gut is 8 to 9 meters long, so this is a massive amount of tissue. So much faster than your skin, than your hair, than any other organ in your body. So when we and this process stops the moment we block the Wnt pathway. And that was, I think, the first link between Wnt signaling and the adult human body, and particularly stem cells. And it's now we know it's not only the gut that listens to Wnt signals, but it's probably all stem cells in the adult are driven by Wnt signals. So this is in like 97, 98. There were no technologies then that would allow us to find out how exactly Wnt would drive these stem cells, particularly because we didn't know the identity of the stem cells. We then had to wait for new technologies. Pat Brown invented for the molecular biologists DNA arraying, which was spectacular, but has meanwhile been replaced by RNA sequence for the molecular biologists. Together with Pat Brown at Stanford, who invented DNA arraying, we essentially we determined what were the genes that this growth factor, Wnt, activated in colon cancer, because we had already also linked it to colon cancer. And are those genes also active in normal crypts? Because crypts are the normal stem cells, cancer are the abnormal stem cells that never stop growing. But it turns out they're very, very similar. Only the cancers don't stop, and the normal stem cells just make just the right number of new daughter cells every day. So using a lot of complicated tricks, we came across And I hope you can see this. I'm not sure if those lights can go off now. So we came across amongst a list of about 60 genes that Pat Brown sent to us. One gene in particular, well, Nick Barker, who is now a professor in Singapore, worked his way through this long list by the technology that were then available. LGR5 looked unique. He created a mouse. We can actually make transgenic mice, knock-in mice, in which we can modify genes in the mouse. And what he did, he modified one of the genes called LGR5, discovered by him, in such a way so he put in a coding sequence that allows the cell that normally activates a gene to emit a green light. Now, what you see here, again, are these villi. I hope you can see them here, 3 villi. This is a real crypt, so it's no longer an animation, it's a real crypt. And these little green fluorescent stripes, each of these is one of these cells that we thought, well, maybe they are the stem cells. They're tiny, so normally you would not have seen them, but now they emit a green light in a live mouse. And as you know, mice typically don't emit light. These ones do, and I'll give you another example. These would be our candidate stem cells. There was also another trick built in that I won't try to explain, but this now allows Nick at will to inject a little bit of tamoxifen in these mice, and he turns these stem cells, these potential stem cells, permanently blue. And if they now make daughter cells, those daughter cells will also be blue. You see that happening here. And as you know, mice are never blue. So what is blue is done by Nick. So we marked this one stem cell in blue, and then the next 2 days you saw these blue daughter cells arising. After 2 days, they start differentiating, maturing into these different cell types. Enterocytes are the cells that take up nutrients. Goblet cells make mucus to let the contents pass a little bit. There are many other cell types. Day 3, 4, and 5, after turning this one cell blue, you see that these blue cells reach the tips of the villi. And 5 days after turning this one stem cell blue, the first blue daughter cells die, exactly as predicted 50 years ago. And if we now don't wait, or if we don't analyze the mice after 5 days, but we wait 2.5 years, you can still see this blue ribbon, implying that the cell that we marked in blue is long-lived, lives as long as a mouse. A mouse lives maximally 3 years. And in that ribbon, you can see what it is capable of producing. And actually, it produces all of the other cells of the gut epithelium. So it's long-lived. It's what we call multipotent. And by our definition, this was the stem cell that we had been looking for 10 years. There was a lot of resistance against this because the textbooks had told us what stem cells should look like, and everything was wrong with these cells. And particularly, there was one thing: they divide every day. And people believed it's a defining characteristic of stem cells that they almost never divide, because when you divide, you can make mistakes. You have to copy your DNA. You can become a cancer cell, for instance. So, but that's very reasonable hypothesis, but these cells divide in the mouse every 23 hours, and they do this lifelong, so about 1,000 times. And they make mistakes, so mice do get gut cancer occasionally, but not very often. So another way of looking at these stem cells, you can see that here. So now Laurens and Hugo, I actually had a meeting with them today. They thought that, well, turning these stem cells blue is a little boring. Why don't we give them different fluorescent colors? And they built this from a rainbow cassette. They built what they call the confetti allele. So now on the simple version, we have 4 colors, but if we cross these mice, we have 10 different colors, and there's about 10 stem cells at the base of a crypt. So we can give them unique colors, and our animator, Ewan Huibe, predicted that this is what we should see. And of course, this looks a little over the top, and we told him. But then we got the real images from these mice. And so if you basically now, rather than turning the stem cells blue, you give them a choice, they pick the colors themselves. But the rest of the lifetime of a mouse, that stem cell will make that color. So these mice have these types of guts. So you see here 3 crypts, so a red crypt, a blue crypt, a yellow crypt, a green crypt. While we're doing this, we realized that this LGR5 gene that we found as a marker for the gut stem cells was essentially a marker for all epithelial stem cells in the entire body of the mouse. So the hairs of these mice also have these beautiful colors. And actually, there was a bet between Laurens and Hugo that Laurens said, if a mouse escapes, you switch off the light, I can find it with the black light. And that's actually because they're beautiful. Every hair has its own color. But we saw the same thing in the stomach epithelium. Other organs, we didn't see much, like liver or pancreas or lung, but the liver and the pancreas and the lung are very stable. They don't turn over rapidly. If they get damaged, all of a sudden the stem cells wake up, but then we can play this trick and then we can see that also in the lung and in the liver, you saw these beautiful colors everywhere. So the fact that these cells were constantly dividing gave us a hard time trying to publish this paper. It was actually rejected twice by Nature before they finally published it. But also when we started thinking, and we were inspired by people who worked with embryonic stem cells already for 20 years, Chie Yamanaka's discovery of the iPS cells, where normal cells could be kept in culture for a long time. There was a strong belief that normal cells cannot grow, they will die in days. And everything we were growing then was a cancer cell. Cell lines for the biologists used extensively in academia and in industry. If you transplant them, they make tumors. They're not normal cells. So I sat down with Toshihiro Sato, now a professor in Tokyo. He was a postdoc in the lab and he had been trying to grow crypts in Tokyo. Didn't really work in 2D on plastic. So we sat down and we had, for about 10 years, after we got interested in how these stem cells work, we had just done random experiments to find out what do these stem cells need. So the stem cells in the gut, what kinds of signals tells them to divide at the exact right speed, and never too much, but also never too little. And we had found out that 3 signals were very important. Wind. Now we actually took a wind activator, R-spondin, epithelial growth factor. The name already tells it. It helps epithelia to grow. And noggin is a BMP inhibitor. So we had 3, and we also knew this from an old mouse experiment that we needed to block BMP. So 3 recombinant proteins. Typically, when you culture cells in a lab, you add serum, cow serum, calf serum. There's no serum in this medium. There is actually Matrigel. This is Minna Bissell has worked on this for 30 years and shown that actually epithelial cells only feel happy when they're inside, when they're touching collagen or similar molecules that are normally present in the body. And Matrigel is a mix of collagen and laminin and other things in 3D. The idea would be that we take one of these stem cells out of the body of the mouse. They emit light at the machines, fax machines that are sorted. So one stem cell, and we expose it to the 3 growth factors in this gel, in this, say, collagen gel, so in 3D. And lo and behold, they started growing. And you can see that here. That was already a big surprise because everybody in the lab before that didn't want this project because they said, this is impossible, I'll waste my postdoc years, et cetera, but they started growing. But there were a number of surprises. First of all, they have never stopped. We still have these cells in culture, and it's now 15 years or so. The telomeres stay long. There was people said no, the telomeres get shorter, the ends of the chromosomes, and the cells will senesce. Never happened. They make structures rather than lumps. And these structures, essentially, when we started looking carefully, and this is Toshi, he first called them mini guts. Now they're called organoids. You asked about the name, I think Minna, or the word organoid has been around for quite some time. And I think Minna has been starting using it for when you take freshly isolated epithelial cells from breast, you put it in Matrigel, they form a structure. I probably had seen that, but I thought that we invented the word, but it was actually much older than that. So you might think this doesn't look like a gut, but then this is a more recent experiment from Norman Sachs. If you put these mini guts into collagen, and collagens are long bundles, so now they line up and you can see they merge, and we can make tubes in a simple Petri dish that are 2-3 centimeters long, and you can see that here, and start looking like a real gut. There's a central lumen that runs all the way across. These little blips are the crypts of Lieberkühn, where normally the stem cells sit, and then you can see this very well, but all of the cell types are there. They're in the right numbers, in the right positions, and the only thing we do, we give collagen, and we give 3 growth factors and nothing else. It's really the stem cells that know extremely well how to build an organ, and that's how your bodies stay intact over 80 or 90 years, because your stem cells in all your different organs, so they only can make their own organ. That's very important. They will never make anything else. Many doctors are transplanting these types of stem cells into organs where they don't belong. They will just die. Or if you're unlucky, they make a tumor. But there might be, I would estimate, maybe 100 to 150 different stem cell types, many still to be discovered, that together maintain your body. Our body is formed from one type of stem cell that's called an embryonic stem cell. And I have one little movie over there. There's, of course, many technologies have been developed over the years that are simple to exploit when you work with cell lines. Organoids are these complex structures in 3D, but eventually our lab and many other labs have contributed technologies. Now we can actually do with organoid cells exactly what we can do with cell lines. And what you see here from a recent paper from two postdocs, Hu and Lin, both are now independent back in China. With CRISPR, they knocked in 3 different colors. Green actually in a marker for goblet cells. They make the slime, the mucus of the gut. You see there's many green cells. Defensin A, the pannate cells, they actually help fight all sorts of bacteria that are in your gut. They are orange. And chromogranin A is a marker for the cells in your gut that make hormones. They're very rare, and there's 6 different types. One of the 6 is called the L cells. That makes GLP-1, and that's this whole hype over Mounjaro and Wegovy and whatever they call these weight loss drugs. Essentially are mimics of a hormone of one of these 6 lineages. And you can see they're all, the purple, the orange, and the green cells are all happily living together in this. This is now a human organoid, and that has been extensively genetically modified. And the modification takes 2 to 3 weeks, so it's very fast. In mice, it would take a year, 2 years to do this, particularly when you want to combine. Now, this is now going back to probably 2011, 2012, there were a lot of people said these cannot be stem cells, these cannot be normal tissue, etc. We sequenced, we never saw cancer mutations. There was one issue that we also afraid of, that we somehow had turned these cells into cancer cells because they kept on growing. And what Toshi did, he was still in my lab, and we collaborated with Mamoru Watanabe in Tokyo, who had set up transplantation and the transplantation approach. And together we did the following experiment. Toshi sorted 1 stem cell out of these mice that Nick made with the light-emitting stem cells. 1 stem cell, Toshi grew it up to about 100 million stem cells. The stem cells are green, but the entire Dutch organoid lines are red, so every cell is fluorescently marked in red. We sent those, we froze them, we sent them to Tokyo. In Tokyo, Mamoru's people had treated mice with chemical DSS, and this creates a colitis type model system. So these mice have ulcers in their large intestine, in their colons. Our collaborators infused these red fluorescent Dutch organoids into the insides of the colons, the large intestines of these mice. They float around. Apparently, this is of course not a real experiment, as some of you might have realized. This is the movie. So, but they as soon as there is exposed damage, they will like attach through the integrins for the insiders, open up, and like a living band-aid, they seal these lesions. And from then on, they actually, this is a real experiment. So here you see one of those colons, large intestines, the large bowel. And here's a patch of Dutch tissue grown from a single stem cell. And actually, Mamoru transplanted about 40 mice with the offspring of 1 stem cell. And these patches persist, so they live as long as the mouse. They never developed into cancers or adenomas, polyps. Also, they never disappeared, because another prediction from the stem cell field was that stem cells that divide too much, they get exhausted and they will die. Also, they never did this. And actually, if you look at the gut under a microscope of a 100-year-old person, the gut still looks the same as a 10-year-old person. The gut doesn't age at all, the insides of the intestine. And by all means, what we could tell from these mice is that it's fully functional. Now, Mamoru has gone on to do human-to-mouse transplants, and now he's currently in a phase 1 trial where he's trying to treat patients with IBD, inflammatory bowel disease, so colitis or Crohn's, that are refractory to the normal treatments. So he can take, what he does, he takes a few biopsies, expands them in 4 times 1 week, 10,000-fold, and then he, with balloons, catheter balloons, he then transplants them back onto the lesions of these patients. And we don't know the outcome, but that, I think, is the first clinical trial of trying to repair tissues with organoids. I told you that LGR5 and these mice showed us that actually these stem cells are everywhere, and the principles of growing these stem cells also seems to work everywhere. So we have, and many other labs now have added protocols. There's always variations on the theme that I showed you. So there's 3 growth factors are always there, but like breast tissue likes estrogen, prostate tissue likes testosterone, liver tissue likes hepatocyte growth factor. So with playing around, it typically takes a new, and there's a lot of postdocs in the audience, it takes, I would say, half a year to a year to optimize a new protocol for a new tissue. And that's when we try to apply it to something, to some medical question or some basic science question, and then publish the protocol with a little scientific story attached. And you can see that, these are very, very different organs, taste buds on your tongue, inner ear, salivary gland. Actually, we're now involved in building corneas, so the clear part of your eye, but also conjunctiva, and going to transplant this hopefully next year in patients in Utrecht. And organoids can be used for very basic science questions, gene function, but they can also be used to build or to model human diseases. And I'll say quite a bit about that if I don't go over time. And that is, we think, very important, because what I also now see in my new job in Basel is that for many diseases, there are no good animal models. Also safety, toxicity, rats and dogs and monkeys, maybe a little bit different, are not really good predictors of how a drug will behave. Will it be curative? So to build human biology in a dish, although very reductionist, so these organoids are not a real liver or they are not a real lung, but they really are better models than what we are currently, than cell lines or than many animal models. And they look really pretty. So here are a few. I think this is airway. If I can recognize all of them, this is liver. This is actually, I'll get back to this, is gut with COVID virus. This is also some virus and some organoid, and this is just a pretty picture. I don't know what it is. And the most extreme one, actually, when COVID happened, I had a few, I don't know if they're here, I can say this, had a few rogue PhD students that came up with some quite spectacular approaches of organoid. They only told me after it worked what they had been doing. And one of them was this. So they had apparently over drinks on a Friday afternoon. They asked, what is the most iconic organ in vertebrates? Because we believe that we can do this in all vertebrates. And they came up with snake venom glands. And then they explained me after the fact that sharks kill about 12 people a year around the globe. So that's 1 per month. And every victim makes it into a world press. Snakes kill about 100,000 to 200,000 people worldwide. They maim another 400,000 or 500,000, losing arms and legs and eyes, and they never make it into the newspaper. And this is also because this doesn't happen on the beaches of Australia, but this happens in India and in the lower income countries. So they thought, well, maybe we can grow snake venom gland organoids, and lo and behold, it works. Using the human growth factors, they make these beautiful snake venom gland. Oh, it's a movie. I didn't know that. Yeah, so they look beautiful. Are they functional? Because there was not much known about snake venom glands. What you see here is the inside. So it's electron microscopy done by my friend Peter Peters in Maastricht. So this gray stuff is the venom. So they make enormous amounts of venom. When you do mass spec, it's real snake venom. These are very strangely modified peptides. You could never make them in other systems. And we could kill lots of things. I mean, they tried it with this. It's not in the paper because I don't think. Then they, of course, because for the molecular biologists here, single cell sequencing was very popular. So without me asking for a budget, it's very expensive. They sent it for single cell sequencing. Then they told me they did, but then I said, well, this is not an annotated genome. So now you have to really work hard, because how do you interpret your results? So then they had to annotate the genome of this particular snake. I don't think the name is here. And we could actually then see, the stem cells had LGR5, snake LGR5, and they listened to the exact same signals that our stem cells in the gut listen to. This is the other technology that, so in this movie that Ian already referred to. So when Toshi in Utrecht did the somatic, the tissue stem cell organoid type, Sasai in Japan came up with a very different approach. So there is a cell, an embryonic stem cell that for me existed 68 years ago for a few days. And they essentially can build my entire body is made by those stem cells, but then I no longer have them. However, Shinya Yamanaka invented a way of taking any cell from your body and basically, in my case, put them back 68 years in time and bring them in that state of an embryonic stem cell that can make everything. And every cell has the full, so my blueprint is in each of my cells. So if I manage to put, and actually you can, well, in my case, it's a little bit hard, you can put a hair out and there's some live cells. Shinya can then turn that into a so-called iPS cell, and those cells can now make every part of my body. Only you have to teach them how to do this. So it's a long, you have to take them to the journey that they normally would go through when I was growing from the one cell fertilized oocyte into a real human body. And so that is a challenge for that other. So we basically start with stem cells already know exactly what they are because they've been through the whole process. iPS cells you have to teach, and these are often half-year-long trajectories. And this is from Jürgen Knoblauch's lab in Vienna, Madeline Lancaster. You saw her picture, did this as a postdoc there, and showing the trajectory of trying to grow human brain from these iPS cells. So in fact, I mean, I could pull a hair out of my head. Shinya will turn into iPS cells. Madeline Lancaster can then make my brain, a little bit of brain out of that. And that's what you see happening here. So they make a little clump of cells. There's different growth factors, different conditions. Typically, the whole procedure, if you do it well, it takes like 6, 7, 8 months. And eventually you get these little lumps. There's also a lot of shaking involved. It's like shaken, not stirred. You see that here. And eventually, what you get is a piece of tissue that has many of the characteristics of the human brain, if you want to do that, or of the human kidney, or of the human skin, or something like that. So this is a different type of organoid. It's a lot harder work to create them. But some tissues we cannot grow, like heart muscle, because heart muscle has no stem cells. The heart doesn't repair itself by creating new muscle. The human brain, there's almost nothing in the human brain that can be repaired. So we cannot grow human brain from an adult brain, but that is actually where you reside on the technology that Sasai originally pioneered. Unfortunately, he passed away about 8 years ago, but that many labs around the world now also are exploiting. So these two go side by side. I think they'll have different applications, and I'll show you some of what we've done with our technology. So one is very early on. This was mostly done by Florijn Dekkers and Jeffrey Beekman's lab in the WKZ with us. Cystic fibrosis. This is a movie I just recently got from them. So essentially it's a disease that hits many organs, but currently is mostly a problem in the lungs. When I did medical school, there were other organs like the pancreas that was involved. Normally in your airways, you constantly secrete chloride ions and water to keep the mucus from becoming viscous. On the right, you see what the CFTR patients have. The CF patients, they lack a gene called CFTR. They cannot keep their mucus in various organs very, very fluid, very liquid. You see the cilia have a hard time moving this tapestry up out of your lungs into your oral cavity. Bacteria grow and you get chronic inflammation. Slowly but steadily, the organ gets destroyed. Now, Vertex in the US had invented 2 small molecules that for many of these patients, not for all, for about half, can correct the function of that gene and can now actually allow the cells to secrete water and keep the mucus a little bit, the slime a little bit more fluid, and then stop the disease process. Miraculous drug. Was actually great for half the patients. So if you look at all of the cystic fibrosis patients, they always have a problem in the same gene, but the mutation can be in many different parts. About half of them have the delta 508 mutation. You see that on the right. So the green part, that's for whom the drug was developed. But there was another 50% that had mutations all over the place, elsewhere in the same gene for that, and they're all rare. They're not like half, but they're like 10 in a particular village somewhere, a small ethnic group or something like that. They could never be in trials, and therefore they would never have access to these drugs. They would not be reimbursed. And of course, it was a new drug, so it was not a cheap drug. So Florijn and Jeff walked into my office one day and said, well, we have an idea. Can we grow from patients that fall in that left category? Can we grow organoids? And we don't have to have lung organoids, but intestinal organoids, because the problem is there as well. And can we then set up a test? And that's where the word avatar comes from. So rather than trying the drug on the patient, which would cost enormous amounts of money and would take a year, can we set up a rapid test of 20 minutes or so, where we basically can see if they do or do not respond. Now, what you see on the left is a normal, so it's a rectal biopsy, it's painless, kids don't really care. In about a week, we can grow these closed rectal organoids, so gut organoids. We open the channel, and you see they rapidly swell. That's what you see in this movie. And that's because water can go into the mini gut, which would be the same as water going into our guts constantly to keep the slime, the mucus, keep it fluid. In the middle, you see what you get with the CF patient. So the organoids grow well, but there's almost no space in the middle, no lumen. And when you open the channel, they don't have it. They don't have a functional channel. They cannot swell. On the right, we treat the organoids in the middle. We treat it with the Vertex compound called Orkambi for a few hours, and we then do the test, and you can now see it looks like the left. So now we basically restored normal function. Now water can go inside these mini guts, and therefore, and when this test is positive, it's now been done for many patients, almost all Dutch patients, as I believe are being tested. When the test is positive, you give the drug to the patient responds. So there's now dozens of Dutch patients who get the drug and get it reimbursed, despite the fact that they were never in this trial that would give them access to the drug. So that's the first time that we've used organoids for personalized medicine. The same thing we can do for cancer. So here the idea would be that here you see this is a man with colon cancer. I think we zoom in. Yeah, so there's the colon cancer. There is in Dutch healthy and diseased tissue. With our technology, we can now grow side by side healthy and diseased tissue from the same patient, and we can have it as live cells in the lab. You see that here. We can sequence their DNA. That's done a lot nowadays for cancers, and see what's wrong in the DNA. And sometimes you can match it with an existing drug to treat the patient. So for that, you don't need to grow the tumors. You could just sequence it straight out of the patient. But more importantly, we have the live cancer cells in our hands in the lab. We can now expose those cancer cells to any drug that exists, and then score what kills the cancer cells and what does not kill the cancer cells. And hopefully we also have the normal cells, and hopefully the drug that kills the cancer cells doesn't kill the normal cells of that patient. Now this has been done now for there's probably about 50 or 60 studies. It turns out that this approach is extremely predictive. On average, 80% of the predictions from the test in the lab on live cancer cells holds up in the clinic. So if the patient is given that drug that kills their cancer cells, it actually works in the clinic very well. This is what they look like. So here you see normal organoids and cancer organoids from the same patient, another one. This is going to be to really now advise the doctors is hard, because for cystic fibrosis, there was only one drug, and the patient either gets the drug or gets nothing. Here, there's, of course, for many cancers, there are really good treatments, and you cannot just go in and say, no, we have a much better idea than the pathologist, etc. You should not give this to your patient, but give that. So there's probably needs industrialization of the assay. It needs to be faster. It's too expensive. You need really trained personnel currently to grow organoids. And there's a number of companies now that try to build little machines, desktop machines, that would allow to do this in maybe say 6 or 7 days rather than the 4, 5, 6 weeks that we currently need. And my belief is here that this will be developed and will nicely complement what we already do for cancer patients. And that would actually make it very similar to antibiotics, where if you have a bacterial infection in your lung, bacteria get cultured in the lab, get exposed to a number of antibiotics. And in the lab, it's seen which one for this patient, which antibiotic will kill their bacteria. And then you basically give that, takes a few days, then you give that to the patient. Here you would do the same, get the cancer cells, go through all your drugs, throw them at the cancer cells, and then decide what works best in the lab, and then advise the oncologist to maybe think about what was seen in the test. Another short story, cancer. So you probably know about the immunotherapies that have been introduced for cancer. So what you see here In yellow on the right are breast cancer organoids. We had at that time about maybe 20 or so lines from 20 different tumors. In light white blue, you see immune cells, so-called CAR T cells. I won't go into too much detail, but you can genetically modify T cells to target them at cancer cells. This was done mostly in Anne Rios's lab with some help from us and Florijn Dekker, but I really liked this study so much. So what they did is this involved a lot of AI coding, but they make movies of about 24 hours where they confront these yellow breast cancer organoids, probably 20, 30, 40 cancer cells per body that you see here, with these T cells that have been modified to kill cancer cells. And then, so what this algorithm does, it basically, these are huge image files. It now tracks individual lymphocytes, these light blue things, the white blood cells here, and in their behavior. And then it classifies the behavior, and you can see there's a few ones that do nothing. They're just lying around. There's some that run around like crazy, but don't seem to kill anything. There's a few that kill, and there's a really rare class that is really good at killing. So with this file, we can now ask the algorithm, show me a typical cell with a typical behavior. And this is a cytotoxic T cell. So this is a lymphocyte that can kill. You can see it's a single cell. It sort of jumps on this organoid. I would estimate maybe 20 cancer cells. You see the clock ticking on the bottom right. It wraps around this organoid. Sometimes pieces break off, but they still keep on killing. And every time something turns red, it is killed. So you see some others that stay nicely yellow. They're not being attacked. But this one is basically, this single T cell kills probably 20 cancer cells in a matter of 24 hours. And I don't think this had ever been seen before. I believe that a T cell that kills can, like a bee, can sting once or twice, but then it dies. But you can see it's still there, still alive, and basically got rid of the entire organoid. And then there is this very small class that in the lab, in Anna's lab, is called the serial killer. It's this one here. So it basically doesn't stay on the outside, but it drills straight through the organoid. Only 2 hours in, probably already killed 10 cells, runs to another organoid. You can see that here, kills a few more here, runs to a third, I believe, in this movie. There it goes. So now 5 hours in the experiment, kills more cells here. Oh, turns around, kills another one, and then disappears into the sunset. It's now only 7 hours into the experiment, killed maybe 30 cells. So these are the most interesting ones to give to patients. So CAR T cells are given to cancer patients as we speak. So to get them, if you spin the organoids down in the centrifuge right now, 2 or 3 hours into the experiment, you have the breast cancer organoids, and these serial killers are inside. We can now analyze what they look like with single cell sequencing. And in this paper that came out last year, we now know exactly what they look like, what markers they have, and this can now be used either to make more of these or maybe sort them out from the 99% of the cells that don't do this, and then give much fewer cells back to patients and cure them. Finally, I have a few examples of how we use this for infectious disease. A very simple one here. This is HSV, herpes simplex virus, that you probably have heard about, and this is one that is yellow, is red. And here you see cervical organoids, so we can actually grow cervical organoids from a PAP brush. And when the virus infects, it multiplies and kills the organoids. And you could use these types of things. I just show this because the colors are nice, but you can use them maybe to develop new antivirals. There are multiple parasites or microbes that cannot be studied in the lab because there's no model system, no cell line that supports the growth of these organoids. One of them is Cryptosporidium. You probably have not heard of it, but it's a malaria-like eukaryote. So it has a real nucleus and real chromosomes, like malaria, as I say. It has a complicated life cycle, like malaria. It doesn't need another vectors, like malaria goes from humans to insects, to mosquitoes, and then back to humans. This one basically goes from mammal to mammal. You can buy the starting stage called the oocyst from a little company in Texas where they basically infect cattle and then they harvest the oocyst. It's like a little egg with which you get infected. And this is a fairly common chronic inflammatory infectious bowel disease, particularly again in low-income countries, but also many in veterinary practices, chickens and cattle, and so can have these infections. And there's no lab model to really study this parasite. So we thought maybe we can solve this in organoids, because cell lines are undifferentiated, they are not the real thing. Maybe what these parasites need to grow is the real thing, the fully mature cells that you would have in a gut organoid. So the idea would be we buy these oocysts from Texas, we inject them into a human organoid that has all of the cell types that it would normally encounter in a real gut. So now it can pick and choose where it wants to adhere, and you see that happening here. So here is the oocyst landing. Inside the oocyst, you might see that there are 4 so-called sporozoites, I believe. You can see them here. They will actually, once it's landed, they will, there it goes, they will actually leave the oocyst, and now they go and look for a cell that they like and where they can infect. Found one, it then forms its first stage, type 1 meront. So it now actually multiplies. It's an amplification step. Again, after a while, about a day, they come out. This is a so-called type 1 merozoite. They again go for looking for cells that they can infect. It turns out they need the enterocytes that are never present in cell lines. These are the cells that take up nutrients in the normal gut. Type 2 meront. So this was the asexual part. Like malaria, there's an asexual and then there's a sexual part. That's happening now. You see they will now form the equivalence of the testis and the ovarium. In front, I think it's the testis equivalent, and in the back, so it's the microgametes. In the back is the ovarium. So there is a single oocyte that needs to be infected. Here they are, the guys. This is the winner. Microgamete, it actually, so it enters forms a zygote, and this now rebuilt that starting point that now will leave the body. I think that will also be visible. And yeah, and there you go. So it's oocyst, it'll leave the body. For cattle, it'll end up on the grass, it'll be eaten up by another animal, and your cycle is complete. So this works very well. This is by scanning EM. We see all of the different stages of this whole cycle inside the organoids, here by transmission EM, all of the different stages that I won't take you through this, but they're quite spectacular forms that they make in a matter of 2 or 3 days. And when we go through the whole cycle, we can actually reinfect mice with what's produced in the organoids. So this is now a platform that's being used by pharmaceutical companies that hope to develop some kind of an antibiotic to fight cryptosporidium. And this was never possible before. A lab in the US, Maria Estes' lab, has done this for norovirus. Norovirus is the virus that on cruise ships or homes of the elderly causes this massive diarrhea and actually kills elderly people. Could never be cultured in the lab. It grows fantastically well in human gut organoids. So again, there's now a platform that can be used to develop antivirals for norovirus. And then finally, when the same group of people that did the snake venom gland also got interested in SARS, and we teamed up with the Corona Lab in Rotterdam, Bart Haagmans and Mart Lamers, you see them here. And we asked, so we were only allowed to do for a while, only allowed to do corona experiments. And of course, we had all these human organoids. It was then, this is March 2020. It was clear it was a pulmonary infection, but there was also indications that other parts of the human body got infected, and we could rapidly test this. And actually, this is a paper that was entirely done and published in 6 weeks, with 1 round of review for the scientists, active scientists here. Tough review, but we managed to do everything. And what you see here is human intestine. We can also do it in lung, but it's great in infecting gut. And in white, you see a single cell that's infected, and by EM you can see the virus comes in from the inside, enters the cell, multiplies, and then leaves the cell on the inside and can only infect other cells in the same organoid, as you can see here. This is 60 hours after infection. And what's white is essentially a viral protein that would normally not be there. We did a lot of molecular stuff, but that was all not very interesting. But then of course, there were all these drugs that people started advertising. Chloroquine was one. Chloroquine is a malaria drug. Works actually in our hands, works exactly as published on cell lines. Cell lines are cancer cells. And the most popular cell line in virology labs is called VeroE6. And VeroE6 is not a human cell line, but it's a monkey kidney cell line. Grows very well, but it's sort of flat, looks like fibroblasts. When you infect, so you have to put a receptor for the virus in, ACE2. If there's no ACE2, it cannot infect, so you have to inspect them. And And when you add chloroquine, you see a dose response curve. At 1 micromolar, it starts killing, and at 10 micromolar, the virus cannot reproduce. So this looks great. If we do that in organoids, and you might realize that chloroquine never worked for patients, actually caused quite some side effects, but never cured patients. In organoids, chloroquine does nothing. 10 micromolar here, the virus happily grows. And there were lots of studies again, and also this is not only our lab, there were other labs that made the same finding. It turns out that in these VeroE6 cells, the virus enters in a very unusual way. It's endocytosed. You see that happening here. So here's the ACE2, the receptor. The virus needs to bind to this molecule. It's normally not there because it's not an epithelial cell. When it binds, it goes inside, ends up in this little vesicle called an endosome, and is finally then releases its RNA, its genome, into the host cell. This is well known to be blocked by chloroquine. So it's no wonder that in the VeroE6 cells, chloroquine was a fantastic drug. But VeroE6 cells do not mimic the insides of your lung or the insides of your gut, because that's where you have fully polarized epithelial cells. They do have ACE2, so you don't have to put it in. But what happens there, the virus actually binds to ACE2, you see that happening here. Then there's another molecule that you only see in epithelial cells on this side of the epithelial cell called TMPRSS2. TMPRSS2 now cleaves the spike protein. That's the component that's actually in the vaccines that we get. It's the outside protein. And this now allows the virus to directly fuse with the host membrane. It never goes inside the cell, the virus particle. It just injects its RNA molecule into the cell. So this is an entirely chloroquine insensitive process. And there's some other drugs that seem to work well. The same thing, they do not block this way of infecting cells. And this would be so in 2020, had the virology labs used organoids rather than cell lines, they would have never proposed that chloroquine would be a good, or hydroxychloroquine would be a good drug for treating COVID patients. With that, I'm a little over time. So I should say in my lab now about 40 years, we probably have had something like 250 young people that have contributed various parts to this story. Of course, there are many other studies that I have not mentioned. A large number of collaborators. There's some here on the slide. And then I want to show, I think some is here. This is actually a funny slide that one of my PhD students showed on his latest presentation. But so with that, thank you very much for your attention. And I hope I left some time for Q&A.
Hans, what a fantastic, inspiring lecture. Thank you very much for sharing such fascinating insights. Yes. For sharing such fascinating insights from the very beginning of your stem cell research in the gut to the actual applications that we see today and which are developing very rapidly today. I think for all of us, your talk and your research work just, reconfirms why we love biology, why we love basic research. Because, starting out with asking a couple of maybe unconventional questions at the time, right, as you explained, over the years, you generate knowledge that then shapes our lives and actually offers a treatment for people in very difficult medical situations. So I think this is clearly a wonderful example for all of us, while science and research is so very important for our future and clearly for our present as well. My name is Agnes Smolensky, and I'm heading EMBO's Science Education and Public Engagement program. And what excites me the most is connecting cutting-edge discoveries with our everyday lives and sharing that excitement with you. And I think you listening to Hans now, that's his lecture sparked a lot of curiosity in all of you. And it definitely changes our perspective as to how we look at research and understand how much it actually affects our lives. It's really a pleasure for me to be moderating the Q&A session for you tonight. And for you sitting in the auditorium, If you have a question, please raise your hand and one of our colleagues will be walking around with a microphone. And of course, for those of you joining us online, we are really thrilled to have you with us today. Please place your questions into the Q&A section on Zoom, and we will make sure that your questions are included in our conversations. And just to give you 2 minutes to think of your own questions, if I may, I would like to ask one question, because I'm sure that our younger viewers would be very, very interested to learn what inspired you to become a scientist. Was there a moment in your life, at some point, which kind of made that click, or a special, yeah?
So my first memory, and this is actually true, is when I moved from Eindhoven to Son in Brabant when I was 5. And so I was not yet 5. Well, that's how I know my memory. That actually at kindergarten, the teachers would stand around me, and then they would ask me, "What do you want to be?" And I said, "I want to be a biologist." And that was because I was always, I learned to read quite early on, and I read lots of books about discovery and biology, but also say the great discoveries in the North and the South Pole and Mount Everest. But eventually I really liked science. And if I can follow it up a little bit, so when I was 18, I went to Utrecht to study biology, and I was extremely disappointed by. There's nobody here from Utrecht now. No, it was then. So we were still in the old buildings in the city. It was all Latin names, taxonomy, anatomy, and there was nothing experimental. It was nothing as exciting as I'd been reading about the discoveries in chemistry and in physics. But then I took up medical school, and I was briefly attracted to be a clinician, a doctor. Eventually, I let that go. Eefkes actually comes from that period. Each other from medical school. But that was when DNA technology came about. And that all of a sudden made biology a really experimental science. And so when our kids were in high school, and I tried to help a little bit with their physics and mathematics, etc., the mathematics and the physics and the chemistry was exactly as I had it at high school. The biology was nothing in those books was known when I did high school. So all of that, the science has come up much, much later, but it's still, every year, I mean, the number of discoveries still are exponential, I would say.
Thank you.
It was a long answer, but more than 2 minutes.
Lovely. It was very, very interesting, I think, for all of us to learn about. Just maybe another piece of information for those of you joining online. If you could maybe add the country where you are joining us from, would be lovely. Would be very, very interesting for us to learn. Okay, then I wonder if there's already a question maybe here in the auditorium. Yes.
Hello, thanks for the nice talk. My name is Laura Wind. I just finished my PhD in Leiden University Medical Center. And I think it's very impressive you already knew at 5 years old that you wanted to become a biologist. For me, it was a bit later, but still pretty young when I was 14 years old when I joined your talk about organoids, also in Amsterdam. You inspired me a lot. And in the end, I worked with organoids. And now I'm actually working on organ-on-chip, specifically tissue engineering of the heart. And I was wondering, if you look at the future, do you see an application of organoids on organ-on-chip, and what would be the benefits and what would be the future of organoids?
Yeah. So I didn't show any organ-on-chip application, but what it is essentially, so these things are pretty unstructured, right? They grow in this jelly stuff. They're all different size, different shape. If you want to be more industrial, if you want to use it in drug development or standardize the assays, you can build a chip around it so that actually you give a shape by the plastic that surrounds the organoid. So we never do that because it takes all the flexibility out of the assay. So what we always want to do new things. If you want to standardize, that is exactly what you need to do. So what I now see, one of my reasons to go to Roche was to see 10, 12 years after organoid technology was first developed, there's now a time, I think it can be applied to a lot of the processes in drug development. And that's exactly where you would need much more standardized, reproducible, statistically significant ways of measuring. So in principle, everything that I showed can be put in an organ on a chip, but I would think it would not be for the type of lab that I try to run in Utrecht. It would be more applied and for industry.
Thank you. I think, yes, there's another question. And there was one, yeah, there was one there. And then let's take yours.
Hello, Mr. Hans. Thank you for the amazing talk. My question is a bit more, how do you say, from background, I studied chemistry. So I spoke to someone from a very big company who works with CAR T-cell therapies. And my question to them was, what do you think about testing on organoids, basically? And the answer was almost biblical. They were like, no, absolutely not. And this particular person was R&D head. So the question is, when we test on organoids, the whole body is like a massive system. Everything is interconnected with each other. So when we do test on organoids, how are we making sure that we are counting in all the factors that can affect to the entire body?
Yeah, so that's definitely not my company. But no, but for instance, CAR T cells, that's typically an example you cannot test in animals because CAR T cells go for human, first of all, the human T cells. If you would inject them into a monkey even, they would immediately be killed. So there is no, so CAR T cells typically you do some measurements and then you can take them straight into patients without much safety data at all. So I would argue actually we in Basel do very similar things. We test them on organoids to be sure that, for instance, they kill the colon cancer, but they do not kill the normal colon epithelium. For other more like small molecule drugs, again, yeah, you're right. So there are scenarios where the drug is taken up by the gut, in the liver is changed into something toxic and then cause a problem in the brain, for instance. You would not find that if you would have the organoid separate. There's a consortium in Holland that actually makes multiple organs on a chip. So they have a gut, liver, brain chip, and you flow the compound through this chip is of course difficult. And then again, what I see of animal experiments, they're really, so everybody always wants to see safety in animals. Often you get very contradicting results from different species. And then still, if everything looks good, you might still run into a surprise if you take it into patients. So what I anticipate is that, so we already do this now with the FDA, that actually we will add it to our package. So we'll have animal experiments and organoid experiments. And slowly but steadily, we will learn where the organoids predict better. And then we might slowly replace them. But so both the industry, but also the regulators are extremely conservative because they know what works and they don't want to invest enormous amounts of money in something that's not really proven 1,000 times over. And the regulatory authorities, they cannot take any risks. So if they would do something new and it goes wrong in patients or in healthy volunteers, and they use a new assay rather than the standard assay, then of course they are to blame heavily. So they're very slow for good reasons to replace what they currently do by something new. So my expectation, as already said, is that we'll see a period where they will be run side by side, and then eventually the better assay will survive.
All right. If you don't mind, I would now take just one or two questions from the online audience, and then we will come back to you. Gabriel from Trieste, from Italy, is asking, how long does it take to personalize a therapy based on organoids on a specific patient with cancer or another disease?
Yeah, so for the example I gave you for cystic fibrosis, but there the situation is quite simple. There's now more drugs, but there was only one drug that you either get or you don't get, and then there is no drug. So that a whole assay can be done in 6-7 days. So basically you get a small piece of tissue, you grow it, and 5, 6, 7 days later you can do the assay. Typically, when we don't speed it up So they don't speed it up. In Jeff Bakeman's lab, would take more like 2 or 3 weeks. But it's not much work, because basically the first day you're busy, and then 6, 7 days later you do the assay. Cancer, because you want to test many more drugs. In the current setting, you need to have a lot more growing, so that takes weeks, 6, 7, 8 weeks, and then you can test 200 drug combinations. But that's too slow, because you usually try to start treatment earlier. And as I said, it's expensive. Also, cystic fibrosis, we have 50 new patients per year in Holland. Cancer patients, we have more than 100,000 per year. So the logistics is enormous, and every hospital needs to be able to do this. So that's where we need the machines, the engineering now to be put in place, if this will ever be applied.
We have another question which is extremely related to this question from Rory Long in Spain. What would be the next step to make organoids even better models to study human disease, in your opinion? What's missing?
Well, for some, cystic fibrosis really works well. And I think cancer works well. There is a group of diseases, chronic inflammatory diseases, like rheumatoid arthritis, SLE, lupus, all sorts of other autoimmune diseases, where there are numerous animal models, multiple sclerosis, but that actually they're caused by some chemical or by some manipulation. They're not caused by the cause of the human disease. They have the symptoms. They're extensively used, inflammatory bowel disease in my field. They have been totally non-predictive. It's clear that they don't tell us what are good drugs. Currently, in many labs, we do a little bit, but much more advanced labs that try to take a human gut, add the human immune system, maybe add the bacteria from the gut, and then build this into a model for human inflammatory bowel disease, colitis or Crohn's, rather than inject a chemical in the mouse and what I showed, which creates something that looks like colitis but isn't colitis. So I think particularly for the chronic inflammatory diseases, also in the brain, diseases like Alzheimer's, Parkinson's or Huntington's or ALS, essentially no good models to develop drugs. I think that's where a lot of work needs to be done, but that's where this technology will likely be unique.
Okay, can we take your question?
Hans, you showed us a lot of wonderful things that can be done with organoids, but I missed the slide with the downsides, the things that you cannot do, the disappointments, the failed experiments, the limits. And I'm specifically asking about that because some outsiders who don't know about organoids think that the organoids will replace animal experiments. Of course, you know that this is nonsense, but I think it should be said clearly, and the downsides should be brought out a little more.
What I did say is that organoids are reductionist. So they are much closer to, well, first of all, they're human, and many of the animal models, of course, are not human. They're closer to the real tissues than are cell lines, for instance, that are now used around the world. They can be made more complex. So people add immune system, people add blood vessels, stroma. There's now integrations of neuronal tissue with other tissues, but they will always be reductionist. So in that sense, they are easier to manipulate. It's much faster, it's cheaper, but it's not an entire organism, and that's very clear. Yeah, but I wouldn't call that a downside. And I must say, as you probably know, when you do understand what you're looking at, almost everything you do in the lab works. And when you're still searching for trying to understand, then almost everything fails. And in labs, you go through long periods where everything fails, right? And it's typically not me, but it's the people in the back there. And but the growing of organoids, I think we cracked that code, because any postdoc in the lab or in other labs who took up a new tissue, in about half a year, you could grow that tissue. And that's then the basis to start studying these. But yeah, of course, it's a reductionist. What you learn, you have to confirm in patients or in animals. Yeah.
Right. Okay. There's a question in the back.
Good evening. I have a question about this organoid can be used to study all kinds of different diseases. And I was wondering, you could study cancer, you could study viruses, but can you also study autoimmune diseases? So your body that attacks your own body.
That's a good question. You have to build them separately. We have done only one disease. Graves' disease is of the thyroid gland, schildklier. There are antibodies that actually activate the TSH receptor, so the receptor for hormone that activates your thyroid. So we can actually, so when we grow thyroid organoids and we take serum from a patient with Hashimoto's with Graves' disease and we add it to the culture, you get an enormous boost of the growth of the organoid. And you can loot it out and that's clear. This is a known mechanism, but we can actually pick that up quite easily. Other labs have taken islet organoids, so islets and pancreas, and confronted them with the T cells or the antibodies from type 1 diabetes patients, and they see killing. So you can build these models, but this often currently takes the form of we already know how the disease works, and now we bring those elements together, and now we reproduce the disease. That would be good enough for drug development, but there's also like rheumatoid arthritis, SLE, IBD, we really don't know the cause. And the question is, can you use these? And you never find this out in animals because they don't have these diseases. So can you actually now find, being guided by what you know from patients, can you find out in these organoids what the real mechanism of the disease is? And as I know, we don't do so much of that, but many labs do this now. And hopefully sooner or later we'll see at least one breakthrough there.
Thank you. Okay. Okay. Now let's take one more question from here.
Good evening, Adam Mistyshin, PhD with Walter Bischmeier at MDC Berlin and previous postdoc at RMCU Utrecht. I have a question related to current attempts to cut scientific granting funds by €1 billion. Your story clearly states that we need basic science for future translational discoveries to treat patients. So what should we do to make proper funding for science, for basic excellent science projects unquestionable in the future?
I think we heard Ian talking about Fotis Kafatos, about how great a lobbyist he was. What I now see, I have seen both sides, academia and industry. So in industry, it's very strongly felt that all breakthroughs come from academia. And academia is not so good in translation. So they really feel that public science should really be focused on basic biology, new technologies, new gene, you name it, and that some translation can be done. But the strong push that I see a lot now, also but everywhere around the world, that you have to be translational. That's because it's typically easier to get nowadays to get money for translational research with an application in mind. But then you sit on the chair of biotechs and of pharma if it's in drug development. So I think that lobby, there's a really key lobby, and Europe is not doing very well there. That basic biology, just the curiosity-driven biology, that is where society benefits most from academia. So I think it's lobbying. Politicians, convince politicians, have some of us sitting in governments helps a bit. But that would be a simple answer. And tell everyone that translation is great, but you need something to translate, right? That's in the word translation. If you have no basic discoveries, there's nothing to translate. And that's often forgotten.
That's a good answer. Thank you.
Okay, now let's take a couple of online questions.
Do you still see Walter Biermeier, or is he? Well, we talk about it over drinks.
Lovely. Okay, then since Yamauri from Switzerland is asking, do you think it will be possible to recreate all the microbiota normally present in the gut?
I see somebody in the back who knows more about it than me. Theo. Yeah, so our approach has been very reductionist there. So the microbiome now is probably involved in a lot of things that have to do with health and disease. It's extremely complex. It differs from day to day, from hour to hour, from individual to individual. So what we have done is take one microbial species and show that it can cause cancer. That would be, again, the reductionist approach. But I know that people are trying to take what they call communities, so groups of bacteria that need each other, that live in our gut, and they try to grow them in organoids. That works as well. But there would be 12, 15 species, not the hundreds or so that are there. Eventually, this is probably possible, but then you essentially are back to a real person, and then you might as well directly study the patient. That's what I would think.
Great. Another question from Muriel from ETH Zurich. How might organoid technology be used to model the interaction between different organ systems, such as gut-brain axis?
Yeah, I already said a little bit. So what's currently being tried, but not by us, is that you have on a chip, you have the different organs you want to connect. And with microfluidics, you can have basically flow from one organ to the next, and you could reverse this. Yeah, that's how that's done.
Okay. Do you have a question here? No, that, okay, I thought, all right, there is.
Hi.
Yeah.
We are high school students, and you just mentioned how the high school subjects of biology was a lot more in depth with your children than it was with when you were a high school student. Do you believe that what you found out should be put into biology books for high school students so that the basic information that we have now, or we found out and the people here are researching, could help future students?
Actually, in
develop
Switzerland, it is. Actually, somebody showed me the high school biology book. Yeah, so I think the basic stem cell biology is very important, and we added some know-how there. Organoids maybe, but then SAS organoids, so the IPS-based organoids, are also going to be very important. But my sense is now, if you look in PubMed, you can look up papers in PubMed and you ask organoids, you can still see every year there are more citations and publications on the subject. So it's being developed as a tool now. It's very common by many, many. So probably belongs in a high school textbook. Yeah.
All right. Thank you. And being mindful of time, I think we would now take one last question from the online audience, which I think very nicely connects to what you just asked, which is from Christopher in Switzerland asking, in the context of organoid research, there is increasing collaboration between universities and large pharmaceutical companies. As a real expert in this field, which environment, academic research or industry, would you recommend to a young scientist starting their career?
I would always start in academia, do a PhD, do a postdoc. You can do a postdoc in a company, but do it in an environment where you're allowed to write papers, and then you can see what you like better. But I don't think you should go very early into industry, because you immediately get focused, and the freedom there is not the same freedom you would find in academia.
Thank you so much. Well, thank you so much for your questions, for the very nice discussion, for your time and participation. And of course, a very, very, very big thank you to Hans for giving such a wonderful talk and taking all our questions and explaining us a lot of very, very exciting topics. And before we close today's lecture, we are really delighted to announce that the next Kafatos lecture will take place next year in March 2025 at the International Student Festival in Trondheim in Norway. And the talk will be given by the Nobel Prize winner, May-Britt Moser. And we would be very happy if you could join us either on site, face-to-face, or online again. Thank you so much for your time. We hope we open new perspectives for you today with the talk and with Hans, and we wish you a very nice evening. Goodbye.